A very good afternoon to one and all. It gives me immense pleasure to welcome you all once again on third day of Faculty Development Program. I, Dr. Pragati Khare, feels privileged to introduce our esteemed resource person for today's FDP on session, Scientific Writing, Publications and Research Funding by Professor Sachin Kumar Singh. Dr. Sachin Kumar Singh is working as professor and head of pharmaceutical chemistry and pharmaceutical analysis at School of Pharmaceutical Sciences, Lovely Professional University, India. He is an honorary professor at School of Medical and Life Sciences, Sunway University, Malaysia. He has 16 years of teaching and research experience. Dr. Singh has completed one DST SERP project on development of S-needs of polypeptide K for treatment of diabetes mellitus. He has also published 700 plus research papers with cumulative impact of 2000 plus. Authored 7 books and 30 plus book chapters. He has 12 granted patents to his credit. He is expert in development of novel drug delivery systems, symbiotics-based formulations, analytical and bioanalytical method development, and BA/BE studies. He has graduated 20 PhD students and 45 master's students, and currently graduating 3 masters and 6 PhD students. He is being featured under top 2% scientists of world as per the list of Stanford University, USA since 2021. Currently, his H index is 72 with 20,000 plus citations. He is a recipient of various national awards, including Dr. B.C. Dev Memorial Award for Popularization of Science 2020, honored by Indian Science Congress Association. He has also awarded Bharat Siksha Ratan Award 2014 by Global Society for Health and Educational Growth, Delhi, and Bharat Gaurav Puruskar 2022 by KTK Outstanding Achievers and Education Foundation, New Delhi, and Distinguished Alumnus Award by Sri Sadganga College of Pharmacy, Karnataka. So I request, sir, please continue with the presentation.
Hello, am I audible?
Sir. Yes, sir.
Yeah, good afternoon, everyone. It's a post-lunch time. I hope I will be able to bear with you and you'll be able to bear with me another 1 hour. So as Madam has well said about that research funding, publications and all. So I am going to cover about research publications and how to write the scientific writing. Research funding I will not cover right now because in 45 minutes to 1 hour that could not be possible to cover 2 domains at a time. So Namaste Satyendra sir. Thank you for hosting this wonderful workshop, inviting me on this platform to talk with your audience and your esteemed faculty members. I can see few are organizing in the IT department also. So let us, without going much deep, let us start about the content of the today's presentation. Please let me know, is it visible to everyone?
Yes, sir. Yes.
Yeah. So let us start. And when we'll go for research methodology, research publications, it is important to understand about why we want to do that. So I will start before going to the main agenda, that what we have to publish and what are the steps for publications of a quality paper. It is important to understand the objective, that why you are in this world of publication. So it becomes important to understand that why the publication has become a main mainstream of the scientific community as well as academic society. If you'll see 10 years back, 10 years back also people were publishing the paper, and what was happening, that those they were getting the awards and all, but now publication has become a mandate for every academic institute. Reason being that now the world, particularly India, has entered into the ranking system. And when India has entered into the ranking system, it became important that every institute should upgrade, the faculty should upgrade themselves, okay, so that they could be able to get something for their institute as well as for themselves. Then comes global recognition. So your publication comes as a passport for you. So what is important in this? The first question is very important: why to publish? Why to publish becomes very important, that it contains 4 different aspects that why you should publish the paper. The first thing is your global visibility. When I am talking about your global visibility, it means if you are publishing, even you don't know anyone in this globe, but when your publications are there, people know you by your name. Not only in this country, but it is in around the globe. Because the person, suppose you are doing research on turmeric, say for example, so whoever so is working on turmeric will be able to understand this fact and will be able to refer your paper when you will go for when you will go for your publications. People who will be working on curcumin, any part of curcumin, when they will see the publications on curcumin, one other person will definitely come to know about this, that this X et al. Team has done this work. Whether you know them or not, they know you or not, but your name will get global visibility. And now it's a time of social media, where LinkedIn is there, where Instagram is there, Facebook is there. There are number of different social media websites where you can find out that if you are doing something positive in terms of achievement, you can also self showcase your publication. That attracts the global partners to become partner with the organization, and that helps in getting the MOUs signed, that helps in getting international collaborations together. So these all are very important for a professor now to become global, that is global visibility. Then comes academic recognition. When you are talking about academic recognition, it means that you are going to be awarded for the hard work which you have done. Nowadays, the universities are having award system, best teacher award, best researcher awards, as well as there are many NGOs who are also applying, means inviting you for giving the award. So those type of, you will become eligible for those type of circumstances where you can get this. Then comes career advancement. Of course, nowadays it is mandatory for getting professorship or directorship or something. Research publication is one of the important parameter which will make your publications in a form of promotions. If you have some stuffs with you, some publications with you, some H index with you, some citations with you, definitely what will happen, that people will get attracted to you. And the profile itself will say, you need not to say who you are, people will automatically know about you, who are you. Then comes research impact and citation. So this is related to your own citation. Now, madam was reading that Dr. Singh is having H index of 72 and citation this much. What is that? That says about your hard work achieved in terms of people who are citing your work. It means that if I have received 20,000 citation, it means that my research work has been cited 20,000 times, at least by some 10,000 people. So I can happily say that at least some 10,000 people know me. That is called research impact and citation. So the question comes, what to publish? Because see, publication does not mean that you have done a data and you are trying to publish the data and the data will go as such. It never happens like this. it is important to understand that what should be your scientific content that should be published. So one thing every time people will say that new and original results or method that advance the knowledge and understanding to the scientific field. Many a time, if you'll go for publication, if you're a beginner of a master students or a PhD student, your teacher will always say, the professor will say, bring the novelty in the work. Bring the novelty in the work. But friends, that's not the true story. Even in the case of patent also, that is not the true story. I'll give very good example of curcumin itself. Curcumin research is going since Ayurvedic time. And research on curcumin is being done today also. Even today also, I can say that one or the two researchers would be working on curcumin in different labs. So see, from Ayurveda ancient time, work is going on curcumin. So why curcumin is not novel? Curcumin is not novel. But the question is that why then 10,000 plus publications are available on curcumin and still publications are coming on curcumin? That is a point to rethink. That what it says that your work should be noble and original, that is one point. But more importantly, how much advancement to the knowledge and science it has given. That is the main step which you should remember. Whatever the work you are doing, you should find out and ensure yourself that is your work is going to provide a new advancement or not. That is important. That whether it is providing a new advancement to your work or not. Then comes reviews or summary of a particular subject or a field. So then you can culminate your things for making a paper. So is it clear what to publish and why to publish? Why to publish will give multiple benefit in terms of ranking, accreditation, promotions, global visibility. But what to publish is something new, or may not be new, but it is providing a new contribution to the scientific community. If something is like that, where you are finding it that what is providing a new contribution to the scientific society, it can be published. So then comes, once you have understood that what is to be published, it becomes important to understand that how to go for what to publish. How will you understand that what is novel for you? What is new, or where the gap is there in the scientific society, the gap which you can fulfill. That is very important to understand. You can see I have shown you some of the journals. Most of the journals are having 10 plus impact factor. These journals are having more than 10 impact factor, and few I can see, Advanced Drug Delivery Review, Fluid and Interface Science, Journal of Control, they are having 20 plus impact factor. So dear friends, when we are talking about such journals where 20 plus impact factor is there, 10 plus, you know what happens? These journals never publish old results or old reviews. They will publish the cut-throat, top-notch articles only, where really the work will be noble, the work will be very much advancing to the society. So if you will read the conclusion and future perspective part of these type of journals, then you will actually able to understand that what is the novelty, because these journals never publish simple review article. They will always take the paper from a very high level scientist who is working continuously in a particular area. And those scientists write the critical review by analyzing pros and cons of that work. So if you read any latest paper of these journals, you will be able to understand that what is the latest trend going nowadays. And based on understanding of your latest trend, you will be able to understand that what new you have to do. So one point I said, how you can understand from the global visibility that what is to be published. So those will be given by the journals, the high impact journals, if you read their conclusion part and future perspective part. Similar way, you have to check for funding agencies also. You have to check for funding agencies also, where you need to understand that how the funding agencies are working. What is the latest first area of the funding agency like DST, ICMR, or international like NIH funding? So these fundings, these communities will always display their thirst area. That current research going in the department is this. They want proposals on this. So that will help you in getting funding opportunities also. So it is important that you should see the top-notch journals as well as the thirst area of different national and international funding agencies and work on that if you really want to have a good publication. Because publications are not as a simply it will come publication, the journal will like to see that how the scientific community will get benefit out of it. If the scientific community is not getting benefit, nowadays there is a SDG, Sustainable Development Goals. There are total 17 goals are there. Out of the 13 are very important goals in which different aspects are there, in which health, pharmacy, medical and health and well-being comes under SDG 3. So under SDG 3, there are many agendas. So if you really want to publish, having an aim to publish is very good. Objective is appreciated, but you have to work for that. Without working, it will not be possible. So before going for making a research topic for yourself, it is important that you should be having a mindset that how I will approach that my paper will get published, or how will I approach that my grant will be sanctioned. Then you should, last two slides I have said what to publish and the way to publish. But there are few things which you should also remember, that what is not to publish, where you should not give effort, because if you give effort, it is a wastage of time. So in that it comes results with lack of scientific interest. If any research which is not providing you scientific benefits to the society or to the any technology, that type of work no good journal will take. Of course, you can do a method of pay and publish. You can pay and you can publish, but that will not work. What will work is you have to bring the contents which are having scientific interest. The second point is outdated work. Any outdated work which have no interest in the public domain. Now, if you'll do lot of work in that area, lot of data also you'll produce, it will not be published because it is an outdated work. So that is why whether you are doing a small work also, economical work also, novelty and scientific community benefit, both are very important. Then do not publish any work which is getting duplicate. What has got published, never publish, because anyway it will not get published. If by mistake it will be published also, it will be retracted. So that is not required. False data you should not publish. Then this is the general guideline on which you can work out that how you can move for the publication part. So this is the body which is related to content, the clear contents, what could be there in your paper. So I hope this is clear, why to publish and what to publish, right?
Yes, sir.
Yeah, fine. So now I will go for making you to understand step by step how your paper will become ornamental so that it can be it can be a very good publication material. So you can see this hierarchy. I have written first introduction, hypothesis, then I have written methods, results, and then I have written discussion, conclusion, title, and abstract. Can anybody say why I have written title and abstract at the last? Whereas in the publication, this title and abstract will be on the first page. So why I have made like this my slide? Anyone? Let us have some interaction also with the audience. Yes, anyone? Any idea why I have made it like this? Opposite? Yes. Any audience?
Any participants can send message or they can unmute yourself. We have open unmute for all.
Okay, no problem. I hope audience is busy. So the problem is that if you start making a title and writing an abstract on the first go itself, it will not work out. Why? Because you are not clear with your introduction. You are not clear with your hypothesis. You are not clear with statistical analysis. You don't be very sure that what will be the best result that you want to write in your abstract. These all things could be possible after drafting a paper only. And that is why I have given first step to introduction, second step to hypothesis, third you should work on method, fourth you should work on result obtained, fifth you should work on discussion, and then finally conclusion. And when everything will be ready, then you will be able to understand that what should be my suitable title that is covering starting from introduction till conclusion. What will be one sentence title that you can take, a simple one sentence title that will be self-explanatory of your work. Is it clear? That is important. Then second thing is abstract. So by the end of your work, discussion and conclusion, you will be very clear that what should be by the important content in the abstract. Because abstract, you have to write very crisp, 200 to 250 words. So you cannot write stories in that. You have to write exactly what is the objective of your work and how you have achieved that. So for that only, title and abstract should be the last part. So how to write a good title? You should avoid writing the technical language. Nowadays, Chat GPT has become a very good friend for everyone. But unfortunately, what is happening? Chat GPT, if anyone is writing from Chat GPT as such, the plagiarism software is catching that, and paper is getting rejected from the desk of the editor itself. It is not going even for peer review. So that is why it is important that you can use Chat GPT to understand the mind mapping that what could be your title. But you should not use any Chat GPT language directly. So what is the best idea? Best idea is that you should write by your own based on your outcomes, your title, and it should be very simple so that it should be easily readable. And unnecessarily avoid jargons, synonymous word, because that will torture your English. So we should not go for getting our tortured English. We should avoid of the jargons. Our sentence of the title must be very clear. The second thing, in the title, your title should be easily searchable. Why we need easily searchable title? Because we need our paper to be cited directly. If you will write anonymous word, people will get confused. If you will write simple, easily searchable title, you know what will happen? On Google, when you will type, anyone will type, your paper will come on first page of that search page. Friends, honestly speaking, nobody looks on the second page of the Google search engine. People only see most of the time the first page of the search engine. People are in so hurry that they don't even bother to go to the second page. I hope you are also doing the same. I'm also mostly trying to do the same. People are not bothered because you know why? Because on Google first page or Science Direct or PubMed first page, whatever will come will be the latest topic or most cited work. That is important. Google will always give, Google is such a search engine that it will always give you the data which is most citable work. Otherwise, Google will not give you anything. So make it easily searchable. Use relevant common keywords. Consult academic librarian if your librarian have expertise, and increase visibility in database like Scopus and Google Scholar. You can increase only which mantra I have given. Keep a very simple keyword, focused keyword. Okay, like you can see functional hyaluronic acid gelatin hydrogel accelerates the closure and healing of diabetic wounds. So in this, hyaluronic acid is one of the important components. So if anyone will type hyaluronic acid, the chances is there that this paper will appear fast. Then substantiate with your data. Spark curiosity. That is very important. The title must be simple, but title must be in such a way that by reading the title itself, people will understand to see that what is written in this. The scientific, like in the morning newspaper. What happens in the morning newspaper? In the morning newspaper, people will try to see that in the morning, what is the hot news? What is the most trending news? Because people will always search which is trending. Curiosity is very important among the public. And dear friends, publication is just like a newspaper. How your newspaper gets published every day, publications also come. So publication becomes important. So if you really want to publish something in a better way, make your title more curious, so that by looking at the title itself, the curiosity should come that I must read the complete paper. I must read the complete paper, which is very important. Then comes the value of your abstract. So friends, your abstract play a very important role in finding the audience who is working suitably in the area of your research. Because if you are making a suitable abstract in you're trying to present, that will improve your followers. That will improve your collaborations. Okay, so if your abstract is written in a well manner, tailored manner, what will happen that the abstract can be converted into a scientific content also. So generally what happens that the structural abstract is there. 1 minute, just a minute. Just a minute, sir. Just a minute. There is nothing happened. No technical issue. I have muted it.
Okay, sir.
Yes, fine. So it comes in such a way that you are having two type of abstract. Example, a structural abstract and non-structured abstract. So the structured abstract, I hope I am audible, right? Hello, am I audible?
Yes, sir. Yes, sir.
Yeah, sorry for just 2 minutes delay because of the some technical official work I was having. Anyway, so there are two things. One is a structured abstract and one is non-structured abstract. Now in the structured abstract, what happened that you have to make a very clear introduction, material methods, results, conclusion, all the thing in a specialized manner. And if you will see the non-structured abstract, you have to write in paragraph. But what is important, what content should be there in the abstract? Your abstract should not be too big that it will take away the readers. Readers should be having interest in your work. So for that, what you have to do that, you have to make a very specific introduction section. You have to make a very specific material. Like you can see, this is a very big introduction. You should not write introduction and back. Introduction should complete in one line, one sentence. Then a scope of the study and background of the work should take 2 lines, 3 lines. That's all sufficient. After that, your objective will come in scope. After that, you should give some 3-4 lines to your methodology and some 4 lines to important results and discussions, and then you should conclude. Ideal abstract should not go more than 200 to 250 words. Rather, I will say that try to complete your abstract within 200 words only. Within 200 words only, we should do it. Otherwise, it is not possible. It will not be a very good abstract. Whether it is one more thing, whether it is a structure, because that is depending upon journal requirement. Some journal says that you clear it, segregate it. This is my introduction, this is my scope, this is my material method, this is my result discussion, and this is my conclusion. Some journal will say give a non-structured abstract in a paragraph. Whatever, whether you are writing a structured abstract or a non-structured abstract, it is very important to make a note that introduction 1 line, scope and background 2 lines, methodology 3 lines, result and discussions 4 lines, and conclusion 1 line. Total 10 to 12 lines it should complete in 200 words. You should not miss this thumb rule. Then becomes keyword. As already I said, your keyword should be very simple, very specific, and easily searchable. Your keyword should be very simple and easily searchable, because your keyword will impact your citations. People will search the article from the keywords only. If keywords are not properly aligned, your article will not be cited properly. Never ever try to give a generalized keyword. Generalized keyword will not work. You have to be very specific in the keyword. So then comes creation of the methodology. Now this is important, that introduction part. So when we are going for writing the introduction part, it becomes important that you should formulate a question. You should formulate a question. To formulate a question, it becomes important that what you want to do the explanation. So what do you want to know or explain is important. Huge observations you have made to write the question that addresses the problem or topic you want to investigate. That is also there. So it is important that what you want to explain is very clear. So this what, who, why, how, when, where are very important. You have to make your introduction more analytical rather than descriptive. More analytical means, we say that like how we do the meshing. You can see I have given lot of things about that, researching the questions. Research, data mine. Before starting the research itself, you try to make it that yes, whatever the thing I am making will be contributing to the scientific community. That is very important. That your work is contributing toward the scientific community. Some novelty is there, some innovativeness is there in your work. So that you have to do research. See, once your objective is clear, once your research question is clear to you, it becomes very easy to do the work. But if you are not clear with your research question itself, and you are confused somewhere in between, then what happens that nothing works. Despite you'll do lot of data, you'll generate lot of data in your lab, it will not work because you have not given time to make a very good hypothesis. And a very good hypothesis can only be created by doing lot of research work, lot of review work, lot of time questioning your work. That is my work noble or not? Is my work scientifically valid or not? Is my work giving knowledge, extra knowledge to the, or extra contribution to the scientific community or not? So these points are very important. Then another point from the student point of view and supervisor point of view also, that whether you have to see your facility also. You can dream that you are going to make a very good nanoparticle for breast cancer therapy and you don't have facility in your lab. Will you be able to do that? It is not possible. So while doing the research work, it is also very important that you have to be very practical. You have to see your own fundings, you have to see your own lab capacity, and you have to work. Your lab capacity is very important, whether it is manageable or not, whether it is within the time frame or not, whether the resources are available or not. That is very important. So manageability is another aspect which should be there. It should be manageable. Then you start doing your work, do a preliminary work, okay? Do a preliminary work and try to do the things, refine the work, try to apply a research method, try to apply a statistics, and then you find out the research method. One thing is very important, that you have to understand your variables, dependent variables. Your research plan must be clear, your steps must be clear, so that every step you can complete in a timeline. Friends, timeline is very important. If you are thinking something, complete in timeline, otherwise nothing will work. Leave about publication, your work itself will not complete. So whenever you are designing something, the timeline becomes very, very important. Then introduction, already I have said you. Literature review also I said you that you have to do enough literature review. I like to say you about the hypothesis also. I said you that you make a hypothesis. Methodology, it is important that in a methodology, replication should be there. Describe the methodology. Replication of methodology is very, very important. Okay, methodology is very important, because what happens that when you are making a methodology, you might get result one time, you might not get the result on the other time. So you have to do multiple experiments while doing this. Then only your research methodology will become reproducible. So your repeatability of the result and validation of the result is very important. So replicate में जरूर करना है आपको study. We have to do the study in replication to see that whether the result is repeatable or not. Because if it is not repeatable, if your paper will get published, and if it is not repeatable, then what the problem will come? The problem will happen that it will lead to retraction of the paper also, and your name will become bad. Then validation will come with a statistical analysis, a standard deviation. You have to find out coefficient of variance, some stats you have to apply. And you can use the softwares also. You can use design expert software. Some very good software also you can use too, like design expert is there, Gastro Plus, because a statistical analysis of your result is very important in order to prove the reproducibility of the work. Without having the mean data, repeatable data, and without having the standard deviation and percentage CB, it will not work. Then comes conclusion. So once your data is ready, then comes analysis of the data, where you have to discuss. In the results already you have got, now you have to discuss with the previously existing work also, that how your work is better or how your work is giving advancement over existing work. That is very important. Existing work, you are not the first person you might be doing that work, someone else have also done. So don't hide that paper, try to cite that paper. Even if you are also repeating some work and trying to advance it, you write in your work that previously we have done this, we have got this result, and this result obtained is much better or much significantly improving the therapeutic outcome than the previously published work, with a proper citation. This is not a negative practice, this is a very good practice. Reviewers will be happy that you have highlighted the previous work and then you are trying to show that you have done something better than previous work. It means you are providing a new knowledge to the scientific community. And once your everything will be, then you go to conclusion. Then you start conclusion. When you will go for conclusion, always remember that you should be very precise. I have seen, I am a reviewer of multiple journal, more than 70-80 journals I used to review. I have seen that many a time conclusion and abstract will be same, which should not be there. Conclusion and abstract should not be same. Conclusion should always be having a very crux report where no result should be discussed. Here you should discuss that what hypothesis you started your work and what is your observations, and then what could be the limitation of your work. You should always mention the limitation of your study, so that any limitation and future perspective, so that if in your lab also anyone is wanting to do the work once again, they should be clear that this much work has been done and this will be the futuristic work. That is very important. And then reference is there. Cite your reference properly. Go try to use reference management software like EndNote, like Mendeley, because it will make your work easier. Suppose your paper has got published and it has not got, means your paper has not got published and getting rejected multiple times. Every time you should not sit and you should start working on the citations. Rather, first time itself you use EndNote to cite your work or Mendeley to cite your work and try to finish it. Because then what will happen, according to journal requirement, you can change the referencing style and it will become easy. The work which has to be done in 5 minutes, it takes 30 hours for a person to do the citations every time if you are doing manually. So try to use some good softwares like EndNote and Mendeley. And then finalize your result and go for publication. Communicate your result, finalize and communicate your result. And once you'll finalize and communicate your result, then wait for the reviewer comments and be very positive in the reviewer comments. So once you have done all the work in a proper way, it could be accepted or it could be rejected. So accepted means enjoy. Your work has been recognized. Your work has been published in a reputed journal. But suppose if not accepted, then it could be rejected or it might get revision, which is quite common. then do not become unhappy. Do not become despair. It happens with everybody. So what you should do, you should be very positive. You should not criticize your work or the reviewer, because they are not your relative or your friends. They are not your enemy. They are no one. They are blind reviewers. Try to find the gap why my paper has got rejected, and appreciate the reviewer comment. Try to improve your work with using reviewer comment, rather than thinking for a 15,000 payment journal and publish in an Indian journal, which is a paid predatory journal. You try to work on the work and try to improve your paper. I am having many examples from my lab. My work has been rejected 7-8 times from even 2-3 impact factor journal. We improved the paper and we published in 15-16 impact factor. There are many examples with me from my lab. So we need some patience. We need some research attitude, which we should understand. We are researcher. The work might be new according to us, but reviewer has found something loophole in that I have to improve that and publish it, improve once again and publish it. So friends, it is very important that you improve, and you can submit to the same journal once again by writing in the cover letter that these were the shortcomings mentioned by the reviewer. We have overcome that shortcoming and we are resubmitting our paper. If the journal does not allow resubmission, then go back to the drawing board. So that is my last, I'm about to conclude, do's and don'ts. Always try to do, first you read the journal guideline. Discussion and conclusion section. Begin with a clear statement of the principal finding. Explain why your outcomes of your study are important to the reader. State whether the result prove or disprove your hypothesis. Introduce new or expanded ways to think about research question. Okay, so this is very important. Okay, the most important thing is that you should always explain that why your outcomes are important for the reader. You should always explain it that why your work should be important. More than do's you already know. Important is don'ts, what you should not do while doing the publication. Do not rewrite your abstract. Always try to use passive voice. Never ever try to have active voice like we investigated, we studied. That's not a right word. We should always write that the research work has been done in the lab. This investigation says about that. Don't suddenly include new argument in your conclusion, which you have never discussed. Suddenly don't bring in the conclusion. Never ever try to apologize. If you are getting limited results also, negative results also, never apologize in your paper. Rather, because limitation is possible with everyone, with every work. It is quite common. So you should work on limitations, never ever apply. It happens with everyone. You should always try to write the limitations of your work, because if you will not write limitation, a new researcher cannot start the work from there. To start the work from there, you should go for understanding the limitation of the study also. And don't unnecessarily overstate your result finding. Okay? Do not overstate your result finding unnecessarily. Do not oversight your things so that it should look that you're too much forcing yourself. So this is all about my thing. So these are some contributions of my research scholars. I am because what they have made me, because of their hard work. These all are the acknowledgements of my research scholars. And these all have not come with 1 day or 2 days. These all are the hard efforts of the scholars, and most of them are, these all whomever I have shown are under the list of top 2% scientists. She has published almost most of her article about 10 impact factor. So that's all from my side. Over to the team.
Thank you, sir, for such an informative session. Sir, one question is from Mukesh Kumar. He is asking that how can we choose the right journal for our research paper?
Yeah, choosing the right journal is very simple. Already I have said, first of all, one has to understand the theme area. So if the theme area is very clear, go to the Google Scholar and write the keyword, and then you can go for journal selection according to your desirability. Once you write, suppose I have to publish on suppose complementary medicine, natural product. So I'll write journals related to nature product. There are websites also like SciFinder and all, from which we can find out. But the best way is that go simply on the Google and then just write your data of interest, title of interest, and you'll get number of journals. And based on that, you can go for selection of the journal. So, Satyendra sir, I have tried to manage myself in a very short span of time, though I would have taken almost 3 hours on that, as you know, because these topics are very practical topics. But I tried to manage it.
Sir, one more question is also there.
I am having my own lecture also, but I will try to find out fast. I am not getting any voice. पूछिए,
Sir, one more question is asked.
Please, please.
How the gaps in research can be identified?
The gap
How
in
the
research,
gaps in research?
yes, the gap can be in research can be identified with very simple thing. Read the high impact journals conclusion. I have written in my PPT also. That high impact journals conclusion will be having a future perspective. So you know what happens? High impact journal always talk about the latest trends which is going on. So that will give a researcher to understand, means the area is very important. You search those journals which are belonging to the area of the researcher. आप अपने journal area में search करें. You have to search it in your area of interest, and then you will find out with latest publication of those journal, because latest publication means recently published. So then only one can understand about the quality of their work, and they can understand the research gap. There is no other way to understand the research gap. Only thing is that read the latest papers published in that area in last 1 month.
Thank you, sir, for the valuable information.
Thank you.
Sir,
Thanks, everyone.
I would like to invite Director sir for vote of thanks.
Good afternoon, everybody present over here in this online session for day 3, session 1. On behalf of BPS Educational Institution of Pharmacy, I extend my heartfelt vote of thanks to our esteemed speaker, Dr. Sachin Kumar Singh, on the session of scientific writing, publishing, and research funding. Sir, your valuable information has cleared all the doubts of the audience. Thank you very much for giving the precious time for this session. Although your session was preponed, it was after 3:15, but due to some technical glitches of resource persons and non-availability, we preponed your session. And thank you very much for this one. And I also, again, thanks to all the participants, those who have participated in this third day, session 1. Thank you very much. Thank you very much, sir.
Thank you, sir. May I leave?
Yes, sure, sir. Sure.
Thank you, sir.
So audience, we will meet again after 15 minutes break. Welcome back again, audience. Coming to the next session, use of data science and data analytics in pharmaceutical science. Dr. Sumit Dwivedi, sir, presently working as Professor, Acropolis Institute of Pharmaceutical Education and Research, Indore, MP. He has completed his B.Pharm from Smriti College of Pharmaceutical Education, Indore, M.Pharm with honors and gold medal in pharmacognosy and phytochemistry from Vinayak Missions University, Salem, Tamil Nadu. PhD from Suresh Gyan Bihar University, Jaipur, Rajasthan. He has been awarded with Fellowship FLSL in 2013 from Pawan Education Society, Gujarat, for his outstanding performance in research on herbal medicine and herbal formulations. He has over 18 years of experience in academics, research, and administration in various pharmacy institutions. With H index 22, has his credit of more than 350 research and review papers in various national and international journals like Elsevier, Science Direct, Wiley, Bentham Science, and Taylor Francis. He has also more than 120 abstracts in various journals of pharmacy and life science. He has published 42 books and serving as executive editor, international member of several international journals like African Journal, American Journal of Pharmacy, Inspirational Teacher Award in 2019. He was also awarded Young Scientist Award in 2018, Young Researcher Award in 2018, Young Achiever Award in 2020, Eminent Researcher and appreciation for DHR Industrial India for adopting his research work formula and methodology formulation of herbal oil and shampoo in November 2018. He has also delivered more than 60 lectures in various conferences and seminars on herbal medicine, standardization of herbal drugs, traditional claims, folklore, uses of herbs and herbal formulations. Dr. Dwivedi has successfully organized 6 seminars funded by MPCST and AERB, workshop on medicinal plants, and completed 2 major research projects funded by Omar Al Mukhtar University, Al Baida, Libya, and Research Cell, OUI, and 1 industrial project. He has also published 25 Indian patent, granted with 1 Indian patent, 1 Australian patent, 22 Indian design patents, 5 UK design, and 4 copyrights also. He has guided more than 35 postgraduate students, awarded 8 PhD, and presently 2 students are working under him for their doctorate degree. He has keen interest in herbal medicine, documentation and validation of formulation, standardization of herbals, cultivation practices of medicinal plants, and phenological studies of herbs. So I once again welcome Dr. Dwivedi. Kindly continue with your presentation.
Thank you so much, Pragati madam. Is I audible?
Yes, sir.
Okay, fine. I have already shared, mailed you my presentation. So, will you please share the same, or shall it? Let me try. Ma'am, can you share the slide? I already mailed you.
Excuse me, sir. Sir, can you present the slide from your side?
Yeah, let me try. It will be just a moment.
कॉल अगेन डेटा आपने जिस व्यक्ति को कॉल किया है वो अन्य कॉल पर व्यस्त है कृपया प्रतीक्षा करें या कुछ समय पश्चात प्रयास करें The person who has called is speaking to someone else. Wait for call again later. आपने जिस व्यक्ति को कॉल किया है वो अन्य कॉल पर
Hello? Hello?
Yes, sir. Yes.
Okay, so the slide is visible?
Yes, sir. Yes.
Okay, fine. Actually, having problem because I am not able to change the slide from here. Pragati madam, is it possible that you can share the slide from your end?
Sir, yes, sir. We are trying. Yes.
Yeah, sure, sure. It's done, right? Okay, so very
Sir.
good evening.
Yes.
So very good evening to one and all, and thank you so much to Dr. Saten Singh sir that they have inviting me for this 5-day FDP on AI. Basically, this topic which was given to me, which I say that which I have chosen is so much close to my heart, and for every pharmaceutical concerned person, because nowadays this AI, data science, or data analytics, they are in the great demand. As far as our newer PCI syllabus is concerned, that from the first semester onwards is focused on these data analytics and data science itself, where the integration of these AI will be there in almost every of the subjects. Okay, so the topic for today's presentation is use of data science and data analytics in pharmaceutical sciences. Moving detail in this particular topic, I would like to say that what exactly these data science and data analytics play a role when we are talking about the pharmaceutical science. Next, please, can you change the slide? So coming to the basic foundation architecture driving between these data science and data analytics, they serves as the foundation for this modern pharmaceutical innovation. Whether we are talking about development of any drug, and to identify the exact gap, and for the identification of that gap, we need to collect certain data, and that data need to be gathered by this data science mechanism only, and finally the gap between raw biological data and the optimized patient outcomes will be highlighted. That is the main reason why these data science and data analytics, they are the prime important in pharmaceutical sciences. Second, the integration of these fields, both data science as well as data analytics, across the drug development cycle, which I am going to tell you later on, that will be basically focused on 4 primary pillars. There are 4 primary pillars. They are started from the transitioning of exploratory data discovery to that of the final post-therapeutic optimization. Now, one question arises, or normally we can say that why these data science as well as data analytics are important to us, or important to any pharmaceutical sciences concerned students or the faculty or anyone who are engaged with this development of the drug. So there are several advantages or the importance of inculcating data science and data analytics in the pharmaceutical sciences or the pharmacy as such. But out of that, the 4 or 5 major concerns are using the implementation of these data science and data analytics. One can accelerate the drug discovery and development, means promising drug candidates that is going to predict the biological activity, and finally they are going to reduce the time and cost of drug development. And one very important, you can see that example of the application of this is we have already seen in our COVID era. Normally, the development of any drug, it will take 20 years from day of initiation. But during COVID, we have already seen that within a 3 or 4 month duration, we had with new drug that is going to treat the new vaccine that is going to treat the coronavirus. So that's because while the development of this drug during implementation of this data science and data analytics, it's become the so much easy or the acceleration in terms of drug discovery and development. Secondly, when we utilize these data analytics and data science together, it will going to optimize the clinical trials results, which can ultimately improve the patient compliance, and they are going to lead more efficient clinical studies. Third, major importance of these include the enhancement of pharmacovigilance and drug safety. Already we have seen that there are several factors by which we are going to increase all these. This is the stages of drug development. When we are talking about a drug development, from the step 1 to step 9, there are various processes that how this drug development occur, which may include the preclinical studies and post-approval studies in between all clinical trial phases. And this entire drug development will be focused on 4 pillars. Next slide, please. That 4 pillars basically indicates that how the from preclinical to the post-approval studies, the development are going to be there. So for that, there are exploratory data discovery, which is considered to be as the first pillar, where the importance of data science came into existence. Means for every raw data that need to be collected, that will be done by exploratory data discovery, which is followed by the clinical data analysis, where the implementation of data analytics came into existence. And after that, both will govern together to form the regulatory submission, followed by the post-market therapeutic optimization. So when the data analysis as well as data analytics, they combine together, they make a distinct primary 4-pillar mechanism by which the entire drug development process will become easier, and it will accelerate in terms of the consumption of the time or some other factors also. Coming to the definition as such, data science, which is defined as an interdisciplinary fields that combine statistics, program, machine learning, and domain knowledge to extract meaningful insight from the data. Means it will be employed for any kinds of interdisciplinary fields. It is not confined to that of the engineering background people or someone who is basically the CSE or IT concerned people. So for that, there are certain components of these data sciences. That components basically includes mathematician, it includes the statistics, programming, machine learning, artificial intelligence, visualization, and the domain expert. Yes, out of all these domain experts play a very vital role whenever the data collection is concerned. But if we have the basic knowledge of mathematics and statistics along with the advanced knowledge of programming to the deeper knowledge of machine learning, and that all we can do with the help of artificial intelligence, then obviously the way by which we are going to collect the data for our study for the drug development become quite easier. Okay, next please. Next parallel approach is the data analytics, which basically defined as the process of examining raw data to the find hidden pathway. Means all the data which you have collected via the gathering of data science or by the utilizing the methods which are adopted for the data from the data science, that need to be examined now, and that examination will be held by the data analytics process. This will draw a very useful condition and helps pupils make better choices. So whenever we are talking about the data analytics process, it basically comprises of 4 or 5 major factors. First was the data collection. That data collection will be done via the data science mechanism. Then that collected data need to be cleaned. Cleaned in the sense, suppose we want to synthesize certain drug, okay, certain API, and we have several synthesis mechanism. So out of that collected mechanism, which is going to reduce the time and give the better yield, that should be taken for the next step. So in this way, the validation, or in this way, the cleaning need to be done. After the cleaning, the third process comes transformation. Yes, out of that 70 or 80 or 100 steps of synthesis, you have cleaned or come to 10 or 5 or 10 methods. So that need to be done, whether they are going to give you the better results or not, or whether the synthesis method approaches or not. And after that, the end product will be done by the analysis. That after analysis, it is going to be the visualization process, where we need to means put the result that the obtained result is up to the mark or not, and finally we have to make the decision. So this is the entire process of data analytics. Next, please. Next slide, please. So these data analytics, they basically are classified into 4 main parameters or the topics, I can say. First among this is the descriptive analytics. Descriptive analysis basically tells that what happened. The third column, key question or answer, that what happens. The major, you can say that the purpose for this type is analysis of historical data to understand what happened by summarizing the trends, patterns, and outcomes. Some examples in terms of pharmaceutical sciences I have kept over here is the analysis of yearly ADR reports of drug from pharmacovigilance database. That is basically mean defined that what exactly happened to the ADR from the pharmacovigilance study. Okay, so that tells you about the descriptive analytics. Next. Next slide, please. Yeah. Second one is the diagnostic analytics, which tells you why did it happen. Okay, so basically means determining reasons for increased incidence of hepatotoxicity associated with drug. So why this hepatotoxicity has been increased? That kind of analytics, when we need to be done, it comes under the diagnostic analysis, which will define you as a evaluation of data to identify the exact cause, relationship, and factor responsible for the observed outcomes. Next. Third is predictive analytics, which basically define that use of statistical models, machine learning, and AI to predict the future outcomes and trends. And it will questions you that what is exactly likely to happen. So, for example, this type of analytics has been utilized in case of drug discovery and development, as well as for the studying of pharmacovigilance, signal detection, and personalized medicine. Some of the examples for in case of pharmacies, AI-based prediction of the patient response to the anti-cancer therapy. And fourth and last type of data analytics is prescriptive analysis, which will tell you what should be done, which basically defined to be as advanced analytics that recommends optimum actions of decision using AI, simulations, and optimization technique. For example, you have all been gone through the optimization of any formulation. So for that, we are using certain design expert software like BBD or this center composite or factorial design. So here, these kinds of analytics need to be done. This is the key summary for all these descriptive, diagnostic, predictive, and prescriptive type of analytics. Next, please. So overall, these all 4 type of analytics will have in linear type of mechanism, you can say that. Firstly, the descriptive, then diagnostic, then predictive, and finally the prescriptive. So means if we say that in descriptive type of analytics, we are focusing that how many customer do I have. So at the final level of data analytics, we came into the conclusion that these customers should get discount to prevent certain conditions. Okay. So in this way, all 4 stages of data analytics works. Whenever we are combining these labels of analytics with the pharmaceutical examples, especially with the drug development. So for the descriptive analysis of existing clinical trials and molecular database to identify the successful drug candidates. For diagnostic, understanding why certain compound failed due to toxicity or due to the pure efficacy. For predictive, what is the implementation, what is the use of AI in the prediction of drug activity, ADMA property and clinical outcomes, whereas for the prescriptive analysis, selection of optimized drug dose as well as the personalized treatment strategies. This is how these analytical levels we are incorporating with our drug development as far as pharmaceutical example of science are concerned. So comparing a major difference between these data science and data analytics, predictive data science, basically it is a predictive, it is based on the machine learning, it is normally will done by the AI based. There are certain intensive programming features which by which these data sciences will govern out, and we can create certain models in case of data science. But coming or talking about the data analytics, they are the descriptive, but they do require certain statistical tools or the analysis. They may be basically born to the dashboard board. They require business intelligence and some kinds of reports we are going to be get generated when we are applying these data analytics to any of the concerned subject, including pharmaceutical sciences. So these are the pharmaceutical data sources, means from where we are going to get data for our study. It may be the clinical trial data, it may be the electronic media record, it may be the genomics, proteomics, drug database, WHO's data, Campbell Drug Bank, then certain devices, hospital information center, wearable devices. These, apart from, they are not the limited. There are certain many more data sources from which where we are going to get data for our pharmaceutical drug development. For example, if we want to work on pharmacovigilance or say ADR monitoring or surveillance of any drug, then obviously we need to go with that of the clinical trials. Coming to, once you have we already have the sources for the drug. Now that drug sources need to be maintain a workflow in data science. So what we need to do? Firstly, you have collected the data. Data collection has been done. Then you need to clean the data. Say you have for ADR monitoring or for the surveillance study for anti-malaria or drug, you have a data of 150 patients. But out of 150 patients, you are not clear that how many of them are suffered from malaria. Okay, so you need to be cleaned. After cleaning, then you need to get integrated that yes, out of 150, only 50 patients are there who are suffering from the malaria which is caused by mosquito, and that will be confirmed or integrated by the blood report test which has been given by the pathology. Finally, the features engineering, then we have to build model building, then we have to validate the all data from data collection to model building, that whether these data are good or means up to date or not. Then we need to get for the visualization. Finally, the decision making should be done. Next slide, please. Yes. So coming to the big data in healthcare, normally there is characteristics 5V rules. 5V rules is volume, velocity, variety, veracity, and values. So volume wise, in healthcare industry, when we are talking about, so all things which are related to the capacity of that concerned pharmaceutical drugs. It should be a transitions, it should be any files, it should be the tetrabytes, that basically concerned with the volume. Veracity is the three major thing, that is the authenticity, availability, and trustworthiness. Means say we want to synthesize any newer drug in our lab, then the person who are engaged in working with us, are they so much authentic? Are they really available? Are they trustworthy to us that they are not going to disclose this formula to anyone? Then coming to the varieties basically structured, unstructured, and multi-factor varieties. Coming to the values, statistical events and correlation. And last one, that is velocity, which basically deals with batch, real time or streams. Some of the examples where in the healthcare sector where these big data mechanism or V3 has been used. Next, previous slide, ma'am, please. Madam, please, yeah, sure. So where these big data has been implemented, they basically focus on genomic sequencing. Genomic sequencing nowadays are in the great demand, means you for any particular disease, say cancer, means in India we are means limited to that of chemotherapy. But in foreign developed country like US, UK, they have created the exact gene which is responsible for causing the cancer. And by the surgery, they are going to change the entire genomic structure of that particular patient. So that all to be done with the help of AI, that all to be done with the help of management or the study of this big data. Second, the imaging, then clinical records, laboratory records, and prescription database. That are some of the limited examples, not limited, but there are some few very major examples for big data in healthcare. Next. Next, please, ma'am. Hello, madam, please do next. Yeah. So once all these data analysis, the different various types of data analysis, but now comes what are the tools for these data analytics by which we are going to collect the information, or the collected information, how we are going to analyze it. So for that, there are four distinct points where these tools need to be classified. First one is the programming. Second one is visualization. Third one is the machine learning. And fourth one is the cloud. So they are the four basic, you can say that classified tools for data analytics. This programming include Python, R, and SQL. They are the very basic programming language, means being in pharmacy or as a pharmaceutical teachers or the students, we also means from the first time already we do have this Python in our new revised syllabus. So we need to be have a basic hands-on training on this, at least Python. And if you are going to use any of the renowned AI database, then obviously within 3 to 7 days you are going to learn that how Python do works. Okay. Coming to the visualization, the three major tools which include Tableau, Power BI, and Excel, out of which Excel we already have familiar with us. Normally, many of us are doing very fluent work in the Excel. Then coming to the machine learning, the TensorFlow, Skitlearn, K-NIM, and Orange, they are the basic analytical tools. And similarly, coming to the cloud, AWS, Google Cloud, and Azure. Out of that, I think Google Cloud, that is very common to all of us. And normally in our Gmail, we are going with the Google Cloud. Apart from these four basic important data analytics tools, some best you can say that some of the more prominent which are used nowadays, which are presented here, like Tableau, Cloudera, and various other tools which basically need to be studied when we are talking about the data analytics tools. Hello, please share the slide. It has been I think Actually, I am not able to change that, so I am unable to share the slide. Yeah. Okay, thank you so much. So, out of all these basic data tools which were being classified in from programming to cloud, these 10 are the most widely used data analytics tools, which include Tableau, Cloudera, Teradata, programming, Spark, Lumifi, Talent, Microsoft, SD Insight, Skyteria, and Pentaho. Next, please. Yes. So now coming to the machine learning fundamentals, since when we are talking about the basic data analytics, first after programming, that programming will basically rely on the machine learning. So that machine learning is classified into 4 or 5 different unit types, out of which first one is supervised learning. And the basic concept of supervised learning is learns from labeled data where input and output are already known. For example, we do have certain readings where we know that what exactly is the input and what is the output we are getting. For example, if we want to plot a graph for a standard curve, we do have the values of the concentration as well as the absorbance. So that comes under your supervised learning. And this particular model, it is trained to predict outcomes based on the previous examples. Some examples, pharmaceutical application includes drug activity prediction, ADMET prediction, disease diagnosis, clinical outcomes prediction. Second type of machine learning is unsupervised learning. This unsupervised learning, it basically finds hidden pattern and relationship is unlabeled data. For example, the model that is going to identify the clusters, similarities, and data structures without predefined outcomes. And they do have a pharmaceutical applications in patient classification, molecular clusterings, as well as in biomarker discoveries. Third major type is the reinforcement learning, where the learns we are going to be have trial and error interaction and improve decision using rewards and penalties. And that is going to select actions to maximize the future rewards. The AI based optimization or the dose adjustment or the clinical studies, they all are focused in reinforcement type of learning in machine learning fundamentals. Next, please. These are some of the common machine learning algorithm. From decision tree to the deep learning, all have the unique principle and function as far as the pharmaceutical applications are concerned. Like decision tree, it uses a tree-like structure to make the decision based on data features. Means the data which are obtained from different sources, they are going to form one umbrella-like structure or tree-like structure. Some very good examples include the drug classification. We have a wider range of drug classification of any category of drug. Similarly, the toxicity predictions or the patient's risk assessment, they comes under the decision algorithm. Second, random forest. Random forest is somehow the combination of multiple decision trees to improve accuracy and reduce error. Means from the decision tree, you have already n number of, or you can say that decision type of structure you have, tree-like structure you that. And from that, you need to find out which are most accurate, first thing, and secondly, how you are going to reduce the errors in it. So for that, random forest type of machine learning algorithm is used. The examples or the application includes your pharmacovigilance signal detections or drug response predictions or some ADMET modeling. Third type of machine learning is support vector machine. Basically, it classifies data by finding the optimum boundary between the different groups. Means, say you have different type of classification of disease, and you need to set certain boundaries or you need to differentiate them. So for that, support vector mechanism type of algorithm you need to be developed. Also, it will be helpful in cancer biomarker identification or molecular activity prediction. Next one is NEP-based, is basically work on the probability-based algorithm which use Bayesian principles for classification. Normally the ADR prediction, text mining, disease diagnosis type of application that are to be done via these NEP vibes. Second last one is the neural mechanism, or sorry, neural networks, which basically mimic human brain neuron to identify the complex data patterns, and that is going to help the drug discovery, image analysis, protein drug interaction prediction, and so on. Last type of algorithm, machine learning algorithm, include deep learning, which include the advanced neural network with multiple layer for large and complex data sets. And here the role of AI-based drug design, molecular structure, precision medicine, imaging, that all comes into the existence. So whenever we are talking about these algorithm, last one is far important because that uses the exact you can say that neural network or the agentic AI that is important for us. Now, next please. Yeah, ma'am. So now the using this data science as well as data analytics, there are so many areas where we can implement all these as far as the pharmaceutical science are concerned. Some of them I have highlighted here in the slides. For example, first one is drug discovery using data sciences. So whenever we need to discover any drug, since in starting slide I have shown you one diagram that from step 1 to step 10, we take almost 20-25 years to develop any drug. And with the help of 4 pillars, that drug need to be developed in various stages from preclinical to post-clinical, utilizing all clinical data. But whenever we are talking about what are the exact, madam, no need to go there, just you, means I was just recalling that slide. But okay, yeah, I was just correlating with that, no need to go there. So that development of the drug from, you can say that from the initiation point to that of the end point, there is a lot of you can say that adjustment a researcher need to be do. Okay, so that all are going to be enhanced when we are using or when we are when we are means incorporating these data sciences as well as data analytics in our discovery process. So there are certain applications like target identification, hit discovery, lead optimization. They all are the pharmaceutical terminologies. Okay, virtual screening, QSAR, molecular docking, ADMET prediction, dose repurposing, all are these prominent apart. They are the prominent areas where we can implement the or incorporate the advantages of these data science, and with the help of data analytical tools we are going to go or enhance or accelerate our drug discovery process. What exactly they are going to do? They are going to give you certain benefits like faster process. One very good example is we have already seen in COVID era. Means in 3 or 4 months, we do have the coronavirus vaccine with us, which normally do require 20-25 years. So that is the implementation of these data science and analytics. They are less expensive. Means all these tools, once you have purchased, it may be used for certain duration of times. And since if you are collecting any data with the softwares, or you are using certain tools, diagnostic tools or analytics tools, then as compared to with that of the human, the chance of error is also very less. So the result you are getting with will be with that of the higher accuracy, right? So this is the one area where we can implement or incorporate the data science and data analytics. Next, please. This is the second area where we need to develop the going for the drug discovery. Again, a diagram is there where so many areas are there, including from epigenetics to the metabolomics, from pharmacophore mapping to drug toxicity to de novo synthesis. These all are the area where we can implement the artificial intelligence, incorporate the sources for the data, and that sources of the data need to be validated with the tools which we are going to study in data analytics for getting the better and efficacious results. So the areas like protein structure predictions, molecular property predictions, biomarker discovery, compound screening, de novo drug synthesis, precise medicine, and not limited to these parameters. We can implement, use the AI in their discovery mechanism. Some examples, very common examples, which will do this for the AlphaFold, DeepMind, in silico medicine, and benevolent AI. They are certain models where we can go for the drug discovery. Next. So into the artificial, because ultimately our aim is to either we are using some question is there. I'll how can AI reduce the time? Yeah, one question is there. Just a minute. How can AI reduce the time and cost of new drug development? Yeah, it's a very good question, and I would like to answer here it right now. Time basically means when we need to develop any drug, we do have we need to do certain if and buts, fit and trial methods. Several permutation combination we need to perform. For example, say I want to assess the analgesic activity of any herbal cream, you say. Okay. So if we use the AI, with the use of AI, we normally have some literature in our mind that whether the set plant, which is extract, which is incorporated in that cream, if it is a single herbal and if it is more than one, so all that have they actually have the analgesic activity or not? So we don't need to perform the individual analgesic activity of individual plants. If it is given and with the help of AI, we can easily find it out, because if we are going to for the manual search, we need to go to the library, we need to search the various journals. Okay, so in this way AI is going to help you to reduce the time. Cost, yes. Why the cost? Because if already the say in vivo analgesic activity of that plants has been reported, so when we have formulated the cream, we don't need to evaluate the analgesic activity of individual plant. So in this way, you are going to reduce certain money also. This is a very means I can say that common example, very layman example. When you correlate with the drug development process, there are various steps where you can, with the use of AI, going to save your time and save your money also. Clear? Next, please. This diagram basically tells about where are the what are the various branches where we can means get use of this AI. Similarly, talking about the ecosystem in pharmaceutical sciences, there is none of the field where we can't implement the AI. Even from the personalized medicine. I don't know how many of you know about the personalized medicine being students, but personalized medicine is the area, or you can say that medicine which is need to be designed according to the patient's need, their age, their sex, their disease profile. So that all is going to be done by this AI apart from clinical trial drug discovery and drug development. Next, please. So once you have gathered all the information, you from the data sciences, you have analyzed with the help of data analytics. Now you need to visualize that data, and that data need to be visualized by so many methods. One very primitive example I am giving about means if pharmaceutics students have worked on design software, they are getting a very good counter plots, colored counter plots. So that is one of the method of data visualization. Coming with the help of data science and data analytics, there are several techniques by which you are going to get the visualization of the data, which include visual elements, some interactives, their techniques, some color plated and designs. There are certain tools by which you can going to create the data in different forms also. Like in Excel, there are different pie chart, bar chart, 2D, 3D. So by this, the data presentation or easily visualization can be done. Next. So when we conclude this implementation or the importance of data sciences, data analytics in combination with that of the pharmaceutical sciences, though there are too many advancements are there in this field, but challenges are at the parallel level. Means I can't say, personally, I can't say that it is very easy job. Implementation of AI in any area of the pharmaceutical science had a great challenge. Like there are certain which I had presented here. That first one is the poor data quality. If we rely on AI always, the data science giving the data to us is of which level we can't, means we need to recheck, re-verify that. Also, certain times some of the data they are missing. Some privacy issues are there. That yes, if some copyright material has been there, whether that need to be given or not, that is also a major concern. Ethical concern is there. Which data need to be provided or which data need not to be given. Lack of skilled workforce, data integration, and finally the regulatory acceptance means which kind of regulatory agencies are going to accept the data which you have generated using AI or by collecting the data from the data sciences, etc. So challenges are at the parallel level, and every individual who are working in it should keep all these challenges in their mind. Next. Coming to the future trends, as far as the pharmaceutical sciences are concerned, these data science as well as data analytics, they are going to play a very pivotal role. And with integration of AI, we are going to have knowledge about, or we are going to tell about this AI, some digital twins, quantum computing, federal learning, blockchain mechanism, some generative AI, which basically collect data from the agentic AIs, LLMs, and precision therapeutics, that all need to be the part of these data sciences and data analytics as far as this pharmaceutical sciences are concerned. And in near future, we all have to accept all these realities. Otherwise, we are not in the race. Next. Next slide, please, madam. Yeah. So for all, I think this is the probably last slide. So for all these integration of data science or data analytics or challenges or the future trend, what we need to be have in our particular, you can say that, mind. So what exactly is required for any personal is that they have a good knowledge of statistics. Excel. In Excel, there are means by the implementation of various formulas, you become the master of statistics. Every formula you are going to incorporate in that Excel sheet. Once you have command on statistics, you need to study about the programming. One basic program, Python, which is already been given in semester first in newer syllabus. And I do learn Python in 7 days, only online video classes. With the use of AI, you just go through the programmings, Pythons. It is very easy to understand. Machine learning, already types and some basic description I have told you. Rather, you have to implement that parameters to in sense of pharmaceutical scientists. Bioinformatics, already the biostat and biological system, NIPER, Hajipur or some more NIPER, they do have MS in bioinformatics. So bioinformatics then, but obvious, without pharmacology, we are nothing. Pharmacology, data visualization, and regulatory knowledge. So these are some of the basic skilled a person need to be inculcated in them so as to better understand the data science, data analytics, AI, and their implementation in pharmaceutical sciences, which is basically more important to know. Next. Thank you so much for all of you. If you have any query, I think one question already I have answered. And yeah, only one question I have answered. If you people have any query, you are feel free to ask. There are, meanwhile, I would like to also state you all that there are certain, you can say that free platforms are available. Free platforms is available in the LLM of Infosys. Last month, I think in May, I visited the Infosys Mangalore campus. They have conducted one 3-day FDP on AI in education. And there they taught us that how with the LLM of Infosys, which is free of cost, and one more classroom is there that is, I think, I'm not remembering the exact name, but it is Skill Bolt is something. Quill Bolt or Skill Bolt. Just a minute. I'm telling you the exact name of that platform which is given in Infosys, and what in that particular domain you are going to assess n number of courses free of charges for the students, for the faculties. They are in Springboard. Yeah, Springboard is there. They are free of it is free of charges, and you may enroll yourself in the courses like generative AI, machine learning courses, LLM, etc. and etc. So yes, do anyone have any query? You are feel free to ask. No questions. No questions means all of you are either so much exhausted or you have gain all the things. I have no doubt. Anyhow, thank you so much. Thank you to BPS College, my dear friend, your principal, Dr. Satyen Singh, sir, for inviting me for this FDP. Thank you so much. सर आपकी आवाज नहीं आ रही है.
Good afternoon, everybody. So, one and all present over here in this 5-day FDP for day 3rd, session 2. I, on this behalf of BPS Educational Institution of Pharmacy, I extend my heartfelt thankful to Dr. Sumit Dwivedi for delivering a lecture on data analytics and data science for the pharmacy. Your valuable information and your clear vision regarding this topic was very clear. It is very informative for all the audience. I am very thankful to you, and I'm also thankful to the participants who joined here for this 5-day FDP. Sir, more than 300 plus 325 or more than around 400 participants joined this FDP program. They have registered around 400 participants.
Good, sir. Good,
So
sir.
from all over India, we got the responses, not only from this north side, we got responses from south side also, east side, west side, everywhere we got the responses. So thank you very much for everybody who made the effort to success this event. Thank you, Dr. Sumit Dwivedi, for
Thank
being
you.
with us. Thank you,
Thank you.
participants.
Thank you so much, sir. Thank you.
Thank you, sir. So record हो गई?